Okay, welcome everybody back to the Helsinki metaproteomics webinar. Today we have Dr. Mohideen Jafari, who is a principal investigator in the Systems Pharmacology Research Group at the University of Helsinki. He also PhD in applied proteomics, and he's also a docent in bioinformatics. So we really look forward to hear you talk about proteins to proteomics today. So please.
Okay.
We'll just switch my video off.
Okay, thank you. Thank you for the invitation and also for the introduction. So today I want to speak about the proteins and proteomics. So you might feel this, it's a little bit basic. So we want to cover some basic concept of proteomics today. And I hope that it would be well aligned with other talks that you have during this week. So during this talk, I will cover some definition that we need to know about them or review them to have a common language. And also, we have a section related to properties of protein. Then we cover some points related to database, protein database. And finally, we have some, the last section is related to MS and MSMS. So what is the difference and what is the application? And also we have two, I can say, assignments or working together or alone during this talk. So hopefully you can find in how to do it. And the first one, it would be in the first slides. So the first question. So we want to review what is the meaning of proteins and proteins and what we want to looking for. So let's see first the results. Okay, so we have it. Most of you answered correctly. So we consider protein as an executive or functional protein, functional molecule inside the biological systems. So the importance of the protein, these kind of molecules, because they are involved in many functions and they are if you exclude the inheritance or regulatory function inside the cell, but most of the function which related from the structural, from the any kind of function related to the cell growth or proliferation are related to the proteins. So let's go to the next second question. So we want to know more about what is the meaning of executive molecule or functional molecule inside the cells. So we know that some functions related to RNA or DNA, but here when we talk about proteins and functions, so we need to clarify it more. So it's nice to see that more students joining. So we have 6 students and then we'll continue after answering. Yes. Yeah, it's very clear and for everyone that, many tasks such as catalysis, we have enzyme, signaling networks, and also transportation. These kind of function mostly executed with molecule or with proteins as executive molecules. So let's go to the third question. Here in this question, I want to highlight a paradox.
Yeah, I said
Okay. So we are waiting for the last student to answer. Yes. Again, most of you correct choose the correct options. And it's very important to see that, that we have gene ontology, we have annotation for the genes, but those annotation, those information comes from the proteins. So not from directly from the sequence of the genes. Many annotations related to molecular function, relate to the cellular component, when the gene is working in which part of the cell, and what is the task of that genes. So we call gene annotation, but actually this annotation comes from the protein study. So that's the importance of the protein to be considered, and proteomics in general. So let's go to the fourth question. So consider that we may have more than one correct option. Okay. I can say the option number C is would be the best definition of the protons in general. But when you go to the literature, you have some definition, such as option number A and B, that when they call the proton of human proton, or when they call them mitochondrial protons, they also use the proton word or terms to describe. But we should consider that the protons differ from the genomics, or proteomics from genomics, since in proteomics we have some dynamic. So it depends on the place and the time that we try to study the proteins. So the protons would be different. So this is important point of proteomics, and also transcriptomics and metabolomics compared to the genomics. So you can say that proteomics is the study of subset of protein present in different parts of the organism, and how they change with the time and varying condition. Thank you so much. Okay, so far we review the proteome definition, but what is the protein? Is it clear for everyone? So let's answer the last question from the first assignment. Yes. Will you join us, or you will?
I'll just stay here in case you need me.
Okay, thank you.
Hello. Here you mean like a functional protein or just a protein?
The protein, just the protein. So you know that in proteomics we would like to identify protein. So what is the protein?
The previous question, I asked another question.
Yes.
Proteins encoded in the genome, then is that the meaning of metaproteome? And if we study about the express protein, which means the proteomics. Am I correct? If I define this as in the metaproteomics, we study the protein encoded in the genome.
No, that's not true.
To my knowledge, the metaproteomics is related to some proteomic study, which is related to the for the microbiome of the, for example, human, or the mixture of the species, from the specific condition. When you consider those kind of proteomics study. It's called metaproteomics. What maybe it has some other definition, I don't know, but not from that side that you mentioned. Okay, we are still waiting for two students to answer. Okay, so see the results. So we need to discuss more about this question. So, I mean, from a biological point of view, maybe you can say that hemoglobin is the one protein, an example. But from the proteomics point of view, when we want to identify protein, nominate the protein in a standard way, and using that information for our identification, then we have a specific definition that if you are familiar with UniProt KB database, we will have some slide related to that database. But the definition that they use is, I mean, the option E. So we don't care protein complex when we are talking about the identifying the proteins. We are consider a subunit that can spend a considerable amount of its lifetime in the cell as a single chain proteins. So the one subunit would be one identity protein ID. So such as this example, you can check it. The alpha subunit and beta subunits are two identity. They have two accession number, two different accession number in UniProt KBs, and then be called as a separate protein. Even from the biological point of view, so these two subunits maybe it's not functional separately, but in a complex, and most of the protein are in the complex, and some of the complex are transient complex, some of them are I mean, established complex, but when we want to talk about the protein, identify the protein and see the number of protein and check the protein identity, the definition that we use is the single chain protein or single chain, not multi-chain or some other part of complex proteins as a protein identity.
Just a small question. Can
Yes.
I ask?
Yeah.
So, when it says heterotetramer, and it says two alpha helices and two beta sheets,
Mhm.
so there are differences between the two alpha helices as well, right? Or are they same subunit? Yeah, the subunit the same.
I will discuss about that matter when we want to discuss about the allelic variant.
Right.
But in general, when we say alpha subunit, it's alpha subunits. So one accession number, one identity. So is there any isomorphism, is there a mutation? That is something else that we will discuss more later. Okay, thank you for the first assignment. Now we have some common language. When I talk about proteomics and then I talk about protein, what I mean. Then let's go back to the slides. Is there any question from the remote student or remote audience? If not, we can continue. Yeah. Okay, so let's categorize proteomics goal. So these three categorization, these three class of proteomics study would be somehow historically also important because, I mean, when we start to study the proteins, first we need to identify the proteins, how many proteins we have, and what is the, and when I say identity of the proteins, I mean the primary structure or the sequence of the amino acids. So the first goal of any proteomics study would be the protein identification. And the next one, we would like to know more attribute or more properties of the protein. And we need to identify more and see what is the function or what is the additional modification and what is the place of the proteins, in location of the protein inside the cell. Those are related to protein characterization. And the last one, the last but not least, it's the protein quantification. After we identify the protein, know what is the function of the protein, In many studies, you would like to compare two different conditions and then see what is the quantification change relatively, and then talk about the disease or any other things, I mean, the variable that we used in our study. So the third category of proteomics study could be a quantification proteomics. So these are somehow we can say three different goals, and they have some, I mean, from the implementation point of view, it has some variety and difference between them. Try to cover some of those. Okay, so I want to discuss about the challenge in protein identification. So we know what is the meaning of proteins, and I mean, in the ideal case, we would like to find a protein that's the unique amino acid sequence, and from the could be as the identity of the proteins, and from the single origin. But as always in biology, we have some exception. So here also we have, for example, in human genome, we have some different genes coded as same proteins, such as histone genes. So we have different genes, but I mean, the sequence of amino acids comes from the different genes. Or we have this similar proteins or very similar proteins from a different origin. So when we identify the proteins from, especially this important in metaproteomics, when we consider a mixture of the species and we want to identify the proteins, and those proteins could come from the bacteria or from the human or some other examples. So in that case, this would be a challenge to identify these proteins related to which species. So that's also the challenge for protein identification. We have also, I mean, another challenge is splice variants. You know that we have many spliceomes, and we have an intron and exon, and sometimes in its protein sequence they call intane and extate. But which is very common in intron and exon in mRNA. So from the same genes, I mean, different combination of the intron removement or exon, then we have a different kind of protein from the same genes. So this is also very important, and I will discuss about that. When we identify proteins, normally we don't identify the whole sequence of the proteins. We identify a peptides from the proteins. Then on that case, maybe we have some challenge to identify which variation of this splicing would be present in our samples. Also, we have allelic variant that you mentioned about the hemoglobin. So in most of the case, I mean, we have 20 to 30% polymorphism. In most of the case, they are not functional. I mean, you cannot discriminate between different allelic variants from a functional point of view. But we have some rare disease that's exactly related to the allelic variant. So identification of those proteins also challenging in protein identification in proteomics. And the last one is PTMs. So you may know that we have more than 500 PTMs, post-translational modification. And identification of those groups inside the protein would be a challenge. So I will discuss later about what kind of the challenge when we have the PTMs or when we want to characterize the PTMs. Okay, so you know that when we call a polypeptide or a single chain proteins, we have a sequence of amino acids that they are I mean, different from the R side group. So we have a C alpha, carbon alpha, carboxylic group, and amine group. And there is a peptide bound between carboxylic group and amine groups. And these bonds called as a backbone. And we have an R side chain which differ the amino acids. So when we want to identify the protein, we want to identify the sequence of the amino acid based on the R side chain. But which attribute can be used to identify the sequence of the proteins or polypeptides or peptides? So before we find the attribute, we need to define that attribute should be, I mean, that attribute should have some criteria to be selected. So here a summary of the criteria to select an attribute. So let's see the first one. So it should be possible to calculate or predict the value from the sequence. So if we have the sequence, then we should find the attribute, I mean, theoretically. So it's very important for us. So it's predictable value. But from the experimental point of view, it should be easily measurable. So if the attribute is very difficult to measure, we need a specific condition or experimental design to measure, that would be very challenging, that it cannot use that attribute for identification. Then it should be, I mean, a very specific attribute. So it shouldn't be, I mean, some attribute that could be assigned to two different sequences of websites. So it would be very specific. If this value is directed to only one polypeptide or proteins. And also it should be constant. I mean, if we want to measure that attribute in different condition or different organism or different situation or different lab situation, it shouldn't be changed. It should be the constant, because it's impossible to control those changes. So here in this table, we can see some of these attributes that we can consider. The first two columns of, I mean, these attributes only for the single amino acids, that we can generalize it to the polypeptides. So the first two is related to the mass. First two columns related to the mass of each molecules. Then pi value and the hydrophobicity index, pK value, and also the occurrence. So the occurrence is not important. I mean, it's just a statistic that based on the available database, how many percent of this amino acid will have in our proteins. So we can see for the pK value, we cannot use pK value because for many amino acids which are not basic or acidic amino acid, we cannot measure the pK value. They don't have the pK value. So this attribute would not be good. And then we have something like hydrophobicity index. So you can see that we have, for example, aspartic acid and also glutamine have the same hydrophobicity index. So this attribute would not be useful to identify the sequence. So we have also PI values, but the PI values have a difficulty to measure in different conditions, and we cannot use this attribute. But I want to consider, pay attention to the mass attribute, which would be constant, measurable, easy measurable using the mass spectrometry. And then we can predict. So if we have a value 156.10, then we can predict that that is which amino acid is here. So it would be arginine. Or if we have 99.30, it would be valine. So then it's predictable. So here we have two different mass. I will discuss soon about them. But I want to just consider, ask you to consider the best attribute that we can use for prediction and identification of the protein would be the mass, because it has all of those criteria that I mentioned. I will discuss about that. So let's try to review some concept related to the mass and from the chemistry to continue. So if you check the literature or using different reference, you will see many times these kind of terms in proteomics study. So many times we use Dalton. So the Dalton definition is, do you know that? It's 1/12 of carbon mass. So this is the the unit that we use to measure of the mass of in proteomics or any other experiment related to mass spectrometry. So we have atomic number, which is equal to the number of proton. So we have isotope. So the isotope, it's those elements that have the same number of proton but different number of neutron. So then they have a similar chemical features but different physical features. So we have mass number, which is the summation of the proton and neutron. Then we have exact mass. So in the case of exact mass, the mass of the lightest isotope of each elements of one molecule, when you consider the summation of those, the lightest one. So you know that for carbon, oxygen, and nitrogen, the lightest isotope is the most abundant one. But in bigger elements, we have a different condition. So we have an exact mass for the lightest isotope, and then we have monoisotopic mass, which is related to the mass of the most abundant isotopes for each element, and then calculated in a molecule or for a peptide or protein. And also we have average mass, sometimes they use. The average mass is the weighted average of the percentage of the different isotope. Then we can sum those values and then get, use the weighted average to measure, calculate the average mass. Also, sometimes you can see that they use nominal mass, which is the round version of the monoisotopic mass. And the difference between monoisotopic mass and the mass number is called as a mass defect. We have, if you are working with gel electrophoresis, so and you use the ladder to identify the size of the proteins, on that time we have an apparent mass. That mass is not accurate. So just it's rough estimation of 150 kilodalton of the proteins. But when you use mass spectrometry, we usually, we call as accurate mass because we measure with the very high resolution methods. And we have also two terms for measuring the errors. So one of them is millimass unit, which is the 1000 Dalton, 0.001 Dalton, it would be 1 MMU to measure the absolute error. So for any methods, we need to identify what is the error. So what is the error rate? And then when you want to identify the protein, you should consider this error rate for the calculation, because we need to consider that this value would not be the exact. It should be a range between based on the error rate that we measured previously. And also we can use ppm, part per million, as a relative error scale. So both of them are used in the proteomics. Okay, let's see for the first example. So when you want to measure the mass of the amino acids, such as valine, so we need to, first decide which kind of mass, monoisotopic mass, average mass, or nominal mass. Then you can see that, for example, we know that this amino acid has 5 carbon, 9 hydrogen, 1 oxygen, and 1 nitrogen. So then for the monoisotopic mass, we use the monoisotopic mass of each element separately and then summation. And for average mass, we also see the average mass of each element and multiply to the number of those elements and then summation. And also for the nominal mass, is based on the number of proton and neutron, or is also the rounded version of the monoisotopic mass. So you can see there is a difference, and when you consider, and the good things from the mass spectrometry is that we can discriminate and distinguish between monoisotopic mass of the valine and average mass of the valine in the one experiment. Or if I want to say more correctly, in the peptides, we can see that the peptides, considering the monoisotopic mass or average mass, So there is a little difference, but we can distinguish between them because of the resolution of the mass spectrometry. But even the mass spectrometry is very high resolution techniques, but it has some error, and we measure the error. So see this example. So imagine that we have measured a protein which is 5 kilodalton, and we know that device or instrument has a error rate of 1 dalton. So it means 1000 MMU. So then if you want to measure the relative error rate or based on ppm, then using this calculation, we can see what is the error rate based on ppm, which is more common to use. So it has 200 ppm error rate, and so then we should consider when we calculate the mass. So during sample preparation in proteomics, we need to treat our samples with different chemical. Sometimes we add some chemical group, then we have an in vitro modification. So when we add some chemical group, then the mass would be changed. And we should consider the change of the mass when we want to identify the peptides. But in many cases, we have in vivo modification, which called as a post-translational modification. So it happens inside the cells. Different, I told you that more than 500 different chemical group could be added to the peptide or sequence. Then we need to consider those. Sometimes we have some cleavage. You know that the pepsin, one of the enzyme which is active when we cleave from the early version of the pepsin. And so some kind of PTM, it's not just adding the one molecule or one group, a chemical group, it would be removing some part of the proteins. So we have many different variety in PTMs, any modification after translation. So it would be very important. And for example, in the case of phosphorylation, imagine that we have a threonine, tyrosine, serine. There are different places that phosphate groups can be connected to the peptide or proteins. And it may have more than one serine in a single peptide, single sequence of the amino acids. And we need to consider that if you want to looking for the phosphorylation, you need to consider adding one group of phosphate, or two group of phosphate, or three, based on the number of sites that it's possible to consider. And then this variety would be very challenging. Also, sometimes we can identify where is the we can identify that one group of phosphate is added to our peptide, but since in that peptide we have more than one serine or more than one tyrosine, we are not sure that where is the location of that, which serine connected to the phosphate. So we need to use some specific method to identify the location of the PTMs. So in addition to the mass spectrometry, using the detected ion of the PTMs, they're also adding any chemical groups to the protein or peptide would have the effect or would be changed the PI of the proteins. And also there are some antibodies to identify specific PTMs on proteins. So there's another example of the PTMs here. So you can see that we have a seven specification for each amino acid. Serine, thyrine, threonine, tyrosine, and other amino acids here. And then when we want to consider the adding phosphate groups, we need to consider the adding this value. So this value is equal to the mass, monaditory mass of the phosphate groups. Then when we want to find a peptide with a specific mass, and we're also interested to see is there any phosphate groups on that samples for that peptides, we need to consider adding this value to make sure about the identification of the PTMs. So far, any question, or is it clear the same? So from the remote audience, if you have a question, we're happy to answer. We are going to the third part of our talk. So here I provide for a very short video that you can make. I mean, it's very helpful to make familiar with the UniProt KB database. So hopefully you can hear from the remote also and also from the audience here.
Welcome to the UniProt tutorial series brought to you by the Genomic Education team at the Jackson Laboratory. This short video provides an introduction to UniProt and demonstrates navigation through the home page. UniProt can be accessed through navigation to the website www.uniprot.org. UniProt stands for the Universal Protein Resource. It is a collaboration between the European Bioinformatics Institute, the SIB Swiss Institute of Bioinformatics, and the Protein Information Resource. This resource is open access, meaning the data is publicly available. UniProt contains information on protein sequences and functions that can be used by scientists or anyone who wants to explore and learn more. On the landing page of UniProt, there is a search bar in the center of the page, which enables you to search the UniProt databases for a protein of interest. Below the search bar are tiles which link to the 4 different databases included in the UniProt resource. The first database listed on the left is the UniProt Knowledge Base, abbreviated UniProt KB. UniProt KB contains functional information about proteins from a wide variety of species, including humans. The second database is the proteomics database, which contains information about sets of proteins expressed within organisms of different species. The third database is the UniProt Reference Clusters database, abbreviated UniRef. UniRef contains data about groups of proteins organized based on the similarity of their protein sequences. The fourth database is the UniProt Archive, abbreviated UniParc. UniParc contains most of the publicly available protein sequence data collected from researchers all over the world. Thank you for listening. To learn more about UniProt and its features, continue to watch the videos in this tutorial series. For other resources from JAX Genomic Education, find us on our website. Welcome to the UniProt tutorial series brought to you by the Genomic Education team at the Jackson Laboratory. This short video will demonstrate how to search for a protein of interest within the UniProt knowledge base, abbreviated UniProt KB. Locating the entry for a specific protein in UniProt KB will enable you to learn more about the structure and function of that protein. To begin, locate the search bar labeled Find Your Protein on the home page of UniProt. You can select which database you would like to search using the drop-down menu on the left of the search bar. For this tutorial, we will search UniProt KB, which is the default option. As an example, we will search for human angiotensin converting enzyme protein, which is abbreviated ACE or ACE. To search for the human ACE protein, type human ACE into the search bar. Then click Search. When the results load, you may see an option to choose between cards or a table to display the results. You can select one by clicking the circle next to the option. If you have used UniProt before, the browser may automatically use a previously selected view option. We will start with the table view by selecting the circle next to Table, and then clicking View Results. At the top of the results page, you will see the name of the database, UniProt KB, along with the number of results based on your search. Below the database name, there is a selection menu for you to be able to switch between card and table view. On the far left are options for further refining your search, including limiting your results to a specific species, or to entries that contain specific types of data or features, such as an enzyme active site. Since we selected the table view, our search results display in a table format. The default table has 6 columns, including entry ID, the entry name, the protein name, the associated gene names, the organism which expresses the protein, and the length of the protein. Each row in the table is a different entry in the UniProt-KB database. To find the correct entry for our protein of interest, we will first check the entry name column. UniProt entry names typically consist of the name of the protein and the name of the organism, styled as protein_organism. So for the human ACE protein, we will locate ACE_human. You can confirm you have found the correct entry by checking that the protein names column reads angiotensin converting enzyme, and that the organism column reads Homo sapiens parentheses human. To access the entry for human ACE from the table view, we would click on the link in the entry column to access the protein entry. Here, the human ACE entry is labeled P12821. Before we navigate to the protein entry, let's also look at the card view. We can switch to the card view by selecting the circle next to cards at the top of the search results. The card view displays each result as an individual card or tile. The title of each card includes the UniProt-KB entry ID and entry name. Below the title is a description which includes the protein name, the associated gene names, the organism which expresses the protein, and the length of the protein. Protein entries which describe an enzyme will also display an enzyme commission or EC identifier. The description also indicates the type of evidence that supports the existence of the protein and provides an annotation score, which is a measure of both the level of detail in the entry and how much experimental evidence exists to support the information in the entry. Below this description are tags, which provide information about the functions of the protein, as well as a summary of the types of data contained within the entry. To find the correct entry for our protein of interest in the card view, we will first check the card title. We will again see the UniProt entry name styled as protein_organism. For the human ACE protein, this displays as ACE_human. You can confirm you have found the correct entry by checking for the protein name and organism in the description below the title, which in this case read angiotensin converting enzyme and Homo sapiens parentheses human. From the card view, we can click on the title, in this case P12821, ACE_human, to access the protein entry. Welcome to the UniProt tutorial series brought to you by the Genomic Education Team at the Jackson Laboratory. This short video will demonstrate how to navigate through the different sections of a UniProt knowledge base, or UniProt-KB, entry. We will begin in the UniProt-KB entry for human angiotensin converting enzyme, which is abbreviated ACE. Each protein entry contains information about protein structure, function, and expression. At the top of each entry is a summary header, which provides basic information about the protein, including its name, length, and associated gene. In this entry, we are looking at angiotensin converting enzyme from humans, which is associated with the gene ACE and is 1,306 amino acids long. Beneath the header is a navigation menu. Tabs on this menu direct to additional features of UniProt that provide information about the selected protein. We will focus on the Entry tab, which is selected by default. Additionally, a clickable table of contents on the left enables you to quickly navigate to different sections of the entry. You can also navigate through the entry by scrolling in your browser. There are many different sections of a UniProt entry. The Function section provides a summary of the known biological functions of the protein, including information about structural features. The Names and Taxonomy section provides protein and gene names, plus information about the organism that is the source of the protein. The subcellular location section provides information about where in a cell the protein can typically be found. The diseases and variants section provides information about any known diseases or phenotypes associated with the protein, along with details on known protein variants. The PTM/processing section provides details on post-translational modification, abbreviated PTM, and processing of the protein. The expression section provides data on the mRNA and protein expression levels within different tissues. The interaction section provides information on quaternary structure and on interactions with other proteins. The structure section provides information on the tertiary structure and secondary structure of the protein. The family and domain section provides information about similarity to other proteins. The sequence and isoform section provides the protein sequence along with any known isoforms or alternate versions of the protein sequence. And finally, the similar proteins section provides links to proteins that are similar in sequence. Thank you for listening. Welcome to the UniProt tutorial series, brought to you by the Genomic Education team at the Jackson Laboratory. This short video will demonstrate how to find protein structure and function information within a UniProt entry. We will begin in the UniProt KB entry for human angiotensin converting enzyme, which is abbreviated ACE. Each protein entry contains information about protein structure, function, and expression. To learn more about the function of our protein of interest, we can navigate to the first section of the main entry page, titled Function. This section contains a summary describing the known functions of the protein. Throughout the description, there are links to references of scientific journal articles and reviews that provide evidence for the functional information. If you would like to learn more, you can click the linked text to be redirected to the relevant article. At the end of the text, there may be tags, such as number of publications or by similarity. These tags indicate that the functional information was derived from a number of scientific papers or based on sequence similarity to a protein in another organism. Clicking a tag provides more information about and links to the relevant sources. Below the initial summary description, there may be additional categories of functional information dependent on the type of protein in the entry and how much is known about the protein. Since ACE is a relatively well-studied enzyme, the entry includes information about catalytic activity, or the chemical reaction that ACE catalyzes, cofactors, or non-protein molecules that are required for an enzyme to be active, activity regulation, or molecules that regulate the enzyme's ability to catalyze reactions, and kinetics, or the speed at which the enzyme catalyzes reactions depending on its environmental conditions. Below all of that information, there is a subsection titled Features. UniProt entries contain information on several different types of protein features, including functional features, post-translational modification sites, and protein variants. Feature information can be accessed either within the relevant sections of the entry or through the feature viewer, which is linked at the top of the page. This tutorial will focus on the functional features displayed within the main UniProt entry. The features subsection within the function section displays information about protein features that are important for the protein's function. First, notice that the length of the protein is shown in gray. For human ACE, this diagram shows amino acid 1 through 1306. Underneath the gray bar, colored shapes identify sections of the protein that are important for its function, also known as functional features. These features are also listed in the table below the gray bar. ACE is an enzyme, so its features include enzymatic active sites. Other features include binding sites, cleavage sites, and other regions of interest. To filter for a particular type of feature, such as the enzyme active site, we can use the Type drop-down menu. This will display all of the relevant amino acid positions for that particular type of feature within the protein. Thank you for listening.
Okay, so you see that our main resource to extract the protein identification sequence, I mean the primary structure, would be UniProt KB. And you know now that we have reviewed entry and unreviewed entry, which is translated from the transcript. And now, if you consider the procedure, the first step to identify the protein is to define and choose the protein database. So from the UniProt KB, most of the time, for example, if you want to focusing study a rat, mouse, or any kind of species, then we need to extract the proteins sequences from the database. So for the protein identification, the first step to define and choose the protein database. So from the protein, from the UniProt KB, we extract those sequences that we are interested. For example, all sequences related to the specific species. Or in some specific study, we can choose some kind of sequence that we are interested to focus. So the first step of the proteomics or protein identification in proteomics is to choosing the database. The second step is to compare the property or the mass from the database, I mean from the theoretical calculation that we can make it based on the database, and also the observed value that we have after the mass spectrometry analysis. So the second step is the matching. So first we need to identify the protein database, then compare and match our observed value with the theoretical value, and this last step is to define a probability of this matching. So we need to follow some statistical models to make sure this matching is not based on the random matching, it would be exact match. So these three steps we can mention as the main steps in proteomics and for protein identification. What is important to consider that when we say we use mass spectrometry to identify proteins, we should consider that we cannot identify proteins or peptides without charge. So in the mass spectrometry analysis, the first step to ionize peptides or proteins, and after adding charge, or in a very rare case, removing charge from the protein and peptide, then that molecule or ion has the ability to flight inside the mass spectrometry. And based on this, I mean, using the different analyzer, we can identify and distinguish between different mass. So if any peptide or proteins cannot be charged for any reason, then we cannot detect it in the mass spectrometry. So one source of missing value is here. that we cannot ionize some peptides or proteins in a proteomics study. But when we add a charge, it means that, for example, if we add one charge, it means we add one proton. So we should consider also the mass of that proton. I mean, and when we want to consider the whole mass, so based on the number of charge, so we have maybe have one charge or more than one charge, then the mass should be calculated based on the number of charge. But the mass is not only the mass of the proteins, we also consider the mass of the charge that is added to the protein or peptides. So now I want to discuss about the two different approaches in proteomics. So one of them is called top-down proteomics. The other one is bottom-up proteomics. If I want to define very briefly what is the difference between these two approaches, we should consider that when we import or when the input of the mass spectrometry device is protein, then we have a top-down proteomics. But when the input, I mean the ion that which is inputted, which we use as the input for the mass spectrometry, if it is the peptide, then we have a bottom-up proteomics. So this is a two different way. So the bottom-up proteomics is more common than top-down proteomics. We have a difficulty to do top-down proteomics. I can say so far we have some studies that claim that they can identify more than 10,000 proteins based on the bottom-up proteomics. But based on the top-down proteomics, I couldn't see any studies more than 1,000 proteins identified using top-down proteomics. So there's a difficulty to do top-down proteomics. Even it has some advantage, but it has several issues that I've summarized here. So you remember that we have an absolute error when we measure something. Any molecule you measure using mass spectrometry, you have an error rate based on your device. And when the molecule is larger, then the error rate is also larger. So one of the issues here. So when you want to measure the very large molecules, you should consider that if you have a error rate, I mean, not very tiny error rate, even if you have a tiny error rate, when the measurement of that molecule is larger, then the error is also larger. So one of the issues to identify, so that we cannot certain about the measurement because the error is larger for that specific values. Another thing that you should consider about the mass distribution. Do you remember that we have different isotopes? So here you can see the sequence, which is very long for the protein, a small protein, but compared to the normal proteomics that in bottom-up proteomics, we have a smaller peptides or a smaller sequence. But in this case, you can see. So do you remember what was the monoisotopic mass? Do you remember the definition? What is the monoisotopic mass? Is the mass of lightest isotope. And what was the most abundant mass?
Most available.
Yes. And what was the average mass? Weighted average of the all isotope. So you can see here that this peak is related to this sequence, the whole peaks. But we don't have one peak. We have a distribution of peaks. And the monoisotopic mass is very little. The intensity, the y-axis is the intensity, and the x-axis is the m/z. So you can see that the monoisotopic mass, which is we are interested to see the monoisotopic mass because it's the most abundant and the lightest. In this case, for the carbon is the light isotope. So we are interested to identify that one, but when we have a very large sequence, the intensity of that molecular mass would be lower than the other type of mass. We have a different peaks which is related to the same sequence, but based on adding considering different isotopes. So for example, here we have 2 or more 3 carbon isotope 13. Here we have 2 carbon and 1 nitrogen. Here, same as like this. And then when you consider where is the most abundant one, which is this mass for the most abundant one. But this mass is not monoisotopic mass. And it's not average mass also. You can see this value, not monoisotopic, not average mass. So, but the average mass is a little bit different, you can see here. So it's very difficult when we have a large sequence, then we want to identify it because we have a mass distribution. We don't have the specific numbers for the specific peptides or specific molecules here. So when we have a larger sequence, we have a difficulty to identify it. So this is another issue that we prefer to do bottom-up proteomics compared to top-down proteomics. When we input the whole protein into the mass spectrometry, then we have a larger or bigger mass distribution for that molecules because it has many carbons, many nitrogen, many oxygen, and then we might have many isotopes of a different combination of the isotope inside that whole sequence. So the next challenge is about some large protein could not be ionized. I mean, some hydrophobic proteins or some proteins, very large proteins, we cannot analyze them even with the top-down proteomics because we cannot ionize them and make them to flight inside the mass spectrometry. Also, the sensitivity in case of proteins is much lower than bottom-up proteomics and in case of peptides. Another main challenge related to modifications. So you remember that we have several serine, several tyrosine, and threonine only for the phosphorylation. And if you want to consider the whole protein sequence, so this number of serine or numbers, the site for phosphorylation, it's exponentially higher and more than when we consider the peptides. So when we have for example, a sequence with 100 amino acid, and inside that we have 20 serine, so the combination of adding phosphate groups only for the serine, it would be much larger than when we consider a peptide with 10 peptides and 2 serines. So you can see that what is the difference in identification. And finally, also making larger scale top-down proteomics is much harder than the bottom-up proteomics if you want to identify more proteins and do in the different conditions. So in general, we prefer to do bottom-up proteomics in many cases, but we have some exception that the researchers try to improve the top-down approach. Okay. So we have, when we use mass spectrometry, we have two different steps. So historically, it's very important that in the first attempt to use a mass spectrometry, we ionized peptides and then detect the mass, and it was finished. I mean, this is the first approach. So if we only use one detection of the mass of the ionized, then if you want to identify the peptide, it's called peptide mass fingerprinting because we only have a value which could be assigned to the whole peptides. So, I mean, nowadays we call the first level of mass or more than first level. So in the case of MSMS, we have 2 levels of mass. So what is the difference between first level and second level? In the first level, we ionize any molecule or peptides and send to the mass spectrometry. There is an analyzer inside the mass, and then detector detect the mass. But if you want to go for the MSMS, we have one more module inside the mass spectrometry called the CID or a fragmentation part. So in that module, we fragment using the atomic bombardment of the ions and produce more ions, which now calls fragment ions. So the first ion that we inputted into the mass, we call parent ions or precursor ions. So when we have a mass of the parent ion or precursor ion, we have the first level of mass spectrometry. But after fragmentation, we have the fragment ion or child ion. So in this case, we have, from this very first ions, then we have a different fragment of that ions, and then again, after fragmentation, the device sends them to the analyzer, and again detection. Then we have a different number which is related to that precursor ion, which is fragmented. And we prefer to fragment from the peptide bond, which would be, if for example, if we have a peptide with 10 different amino acids and then fragmented, then we have a series of the ideal case. We have a fragment with 9, fragment with 8, 7, 6, 5, 4, 3, 2, and 1 amino acids. So then the difference between the fragment would be equal to the 1 amino acid. Then we can identify the whole peptides. So when we just focusing on doing fingerprinting mass spectrometry, then normally our samples shouldn't be very complex. So we have a protein separation using, for example, gel electrophoresis, and then in-gel digestion, one bond from the gel electrophoresis, and then digested to peptide, and again separate the peptide and use the mass spectrometry. And I can say historically this way it was very common, but nowadays most of the works and high throughput proteomics, bottom-up proteomics use the MSMS to make sure about the sequence. In that case, we cannot make sure about the sequence as I told you on the whiteboard. But yes, this is the difference, that we try to first digest the whole samples and then try to use different method to fractionate and separate peptides. And it's very important a step before mass spectrometry, because if our sample is complex and we want to send the whole samples into mass spectrometry, so there is a co-elution problem. So we try to use fractionation method before mass spectrometry to differentiate from different point of view. For example, if we use the reverse phase columns as a chromatography, so we try to differentiate peptide based on the hydrophobicity, and it would be the orthogonal attribute to separate peptides, not only based on the mass. Also use this feature or attribute of the proteins to differentiate between them. So the last part that I want to explain is related to the database that we have. So we have a database. We know we select all, for example, E. coli proteins from the UniProt. And then we have also observation. So we need a calculation to see which kind of peptide we would have if we used any specific protease like trypsin. So in the ideal case, we should have the peptide after digestion. The experimental peptide and theoretical peptide should be the same. But in reality, it's not like that. So imagine we have the samples of the E. coli, and then we use the protease, trypsin, and then we have several peptides. For each protein, for example, on average, we have 5 to 20 different peptides. And then we measure those peptides. But there is some difference between our calculation and our measurement. So one of the source of the difference is related to the contamination. So you have some, even you are working on E. coli proteins, but you cannot avoid many contamination of the humans, such as keratin. So we have some peptide related to the keratin, which you don't, you're not interested to study them. So we have, that one of the source of difference between the experimental and theoretical is related to contaminant. The other source is related to the, modifications. So when you have the different modifications and you didn't consider those modifications, so if you are not if you don't have any hypothesis related to the phosphorylation or acetylation or any other kind of PTMs, then you don't consider those mass values in your theoretical calculation. So if you put those or you don't put those, so there is a difference between your peptide with modification in experimental samples and your theoretical peptides that you measure, that you calculated. So it depends. If you want to put all possible modification, then the search space would be very large and it takes very large time to find. If you remove all of those, so it means that you miss some information. So if you have some purpose to include some of the PTMs, that would be important. And also you have a miscleavage. So when we, when we want to digest protein, we use the enzyme. And you know that every biological entity have some error. So one of the error from the protease, or such as trypsin, is, for example, trypsin should cleave every lysine or arginine, but in some case, because of the many reasons, many chemical reason or biochemical reason, miss one lysine. And then what's happened? For example, if we have 3 lysine inside the protein and miss one lysine, then we have a larger peptide, including lysine. So it was, it could be calculated. You can calculate it. For example, consider 1 miscleavage or 2 miscleavage. Even, the more is not justifiable that you have a very bad enzyme that has lots of miscleavage, but 1 or 2 it's common to consider. So you may have those peptides in your experimental samples and observation, and you also you can calculate it and put, for example, 1 or 2. But if you have more than 1 or 2, then you cannot match between theoretical and experimental. So these are the difference between the peptides that we have after using digestion, using such as trypsin, and also from the database that we should consider. And one thing more about the PUT, we can calculate all possible peptides from the database, but not all peptide could be observed in the reality. So we have some another resource that we cannot match all theoretical peptides, different data, that we cannot measure those peptides for any reason, for coevolution problem, for not ionization, or for any other problem that would have, and it happens to not have the match between the theoretical and experimental. Okay, so there's a methods to mention what is the cleavage site of different protocols. So nowadays trypsin is more common, but you should consider that we have some other protocols, and which is sometimes we use them. It's not very rare, but in some case we use those protocols that you can see here. So if you want to make sure that, so for the calculation, we need to make sure that where is the cleavage site and where is the cleavage point, to see what is the peptide length, and then calculate the mass. So we need to know what is the logic behind each protease to have a digestion. So for example, in trypsin, we can see that based on this nomination that they use inside the crochet, they use R and K. It means that after arginine or lysine, we have a cleavage point, which point mentioned, and if it is not proline after that, so then trypsin would work. So this is a method that you can use for theoretical calculation of the peptide after digestion. But for other enzymes, it would be different. So we have a lysine C, which is also common, which only work after lysine, and there is no any amino acid to prevent this digestion. But we have some proteins such as pepsin, as you can see here, which have two different, if it works in pH 1.3, I'm here inside our stomach, it's different, different method of cleavage that you can see here. So it shouldn't be any kind of this amino acid before this cleavage site, and also not protein after that. Then we have a different peptide. So you can see and we have a different protols with different method of cleavage, and it's important to choose which one. So in most of the case we use trypsin. So we have some reason to use trypsin, such as the trypsin is very specific, even if you consider one or two miscleavage, but after lysine and arginine, so its calculation is not complicated such as pepsin. And also it's available because nowadays we have lots of pure trypsin, recombinant trypsin, we can use it, and so we don't need to worry about the purity of those protols. And in many experimental setup we can use them, and one more positive thing about trypsin is those peptides that have a lysine, because every peptide has lysine or arginine. So then the ionization of those peptides is easier than if it has some other non-acidic or non-basic amino acids in the inside the N-terminus or C-terminus of the peptides. Yeah, but sometimes we use some other protocols such as lysine C or chymotrypsin, especially when you want to focus on the specific protein and you want to increase the coverage of the identification. So normally, if we have 2, 3, 4 peptides, which is covered around 20% to 40% of the protein, we have a good score of the identification. But if you want to focus on the different isoform of protein, and you want to focus on the allelic variant, or to know the whole sequence more specifically, then it's better to use different combination of the proteins to have a mixture of the peptide, which those are overlap to each other. And then you can make sure about the sequence and the coverage of the sequence in general.
I just had a small question in the cleavage section.
Yes.
So, in this slide, it seems like most of the enzymes don't work if there is a proline group.
Yes.
Do we know why? Why does proline not make them work?
It has some related to the conformational change of the proline, because the proline has some many loops inside the protein. The structure, 3D structure of the protein comes from the proline. So the proline changed the conformation that our enzyme cannot work, but I don't know exactly what is the reason. For each enzyme we need to consider.
There's so many enzymes and all of them don't work if there is a protein present.
Yeah, and the proline is interesting amino acid in proteomics, that and also we have one software called proline to identify the proteins. Yeah, the name they use the protein for as a name. So it's interesting amino acid in proteomics in general. So, and this is the last slide. And in this slide, I want to just highlight the importance of the peptides. Even we say that we are study proteins, and this is a proteomics, but from the implementation point of view, we are study peptides. We need to digest the peptide. It's difficult to do top-down proteomics, so we prefer to do bottom-up proteomics, and we try to identify peptides. And from each protein, we cannot identify the whole peptide, the whole sequence. The coverage near to 100 is very rare in some specific case. In most of the case, around 20 to 40% coverage would be enough for us to identify the proteins. And that one is based on the peptide. So we fractionate peptide, we identify peptide, and then we fragment peptide to different fragment ions. And we are working on peptide to identify, to do proteomics. And it was very, very important to pay attention. Yes. Okay, then, if you have any question, I'm happy to answer, but please, again, go to the Go Socrative and give me feedback before the end of the meetings. But meanwhile, I am happy to answer any question that you want to ask. And let me open the one of the feedback forms. Finish this one. Need to put your name. But meanwhile, if you have any question from the remote audience or
Yeah, thanks a lot, Mohi.
welcome.
I think it was very valuable, at least to us, because we have kind of just ordered services from the proteomics core facility and kind of jumping directly to metaproteomics without really spending too much time on understanding the fundamental basics of the method. So it's good to do it now. Maybe we should have done it in the beginning, but better late than never. Maybe a couple of questions. Something that it's maybe kind of a little bit specific, perhaps, and let me know if it's like takes too long to explain. But something that I didn't fully understand. So data-dependent acquisition and data-independent acquisition. So I didn't fully understand how
Actually, I didn't
that
mention that.
works.
Yeah.
Is it possible to explain that
Yes,
like
yes.
briefly?
And so, if you remember that when I discussed about fragmentation, so I mean, in the first level of mass spectrometry, we have several peaks in each second in the spectrum, and
Yeah.
we need to choose from those peaks to send it for the fragmentation, and then we have a second level of mass spectrometry.
Okay. So it doesn't analyze everything.
Yes. In DDA method, we don't analyze everything. And then the normal, I mean, the typical method is to choose top n peak with consider the intensity. For example, top 7 peak with high intensity selected to go to the fragmentation in turns and then detect the fragment ions. But in DIA method, which is recently more common and many groups working on that, they sent all peaks in the groups. I mean, don't care about the intensity. So they send all peaks to the fragmentation and have a fragment ions. So in this case, we have a more complex data, and nowadays, because of the many methods which can better working on the identification of the different fragment ions, the DI is more common, but even it has some limitation in some case. But I mean, nowadays DI is more common because we don't ignore or miss some of the parent ion or precursor ion to be fragmented and identify them. So we have more data to be analyzed, and there is some specific model using for identification in that case. But the main difference between DDA and DIA, in DDA, which is data dependent acquisition, we based on the data, I mean the intensity data, we choose some of the peptides, some of the parent ion or precursor ion for fragmentation. But in DIA, it is independent from the intensity. So we send all of the parent ions to the fragmentation in it. I mean, not all of them at the same times, but in a different groups. But at the end, all of them will be fragmented